cerebrospinal fluid, or CSF, isn't just housed within this subarachnoid space. We've got to ask, where does it come from and how does it end up getting to the subarachnoid space and why is it there? And for this purpose, we're going to look at the ventricles. So the ventricles are essentially spaces in the brain that produce and contain cerebrospinal fluid. So let's first look at cerebrospinal fluid production, CSF production. So where is it produced? Well, it's produced in an area called the choroid plexus. And the choroid plexus is located within the ventricles, which is these areas that you can see here that are highlighted in blue here. And what CSF essentially is, the contents of CSF, is just like an ultrafiltrate of blood. So all the big stuff in blood has been filtered out. So large plasma proteins have been filtered out. There's no red blood cells in cerebrospinal fluid. We're just kind of left with glucose, electrolytes, and small proteins. What are the functions of the CSF? Well, there are many, but the most important one is going to be buoyancy. You see, the brain weighs, on average, about 1,500 grams. But because we have this cerebrospinal fluid surrounding the brain, what it means is that it keeps it buoyant. If the brain didn't have cerebrospinal fluid, then it would simply be crushed under the weight of its own mass. And so because it's buoyant, it means that it doesn't crush itself, it doesn't compress itself. There are a few things that nerves really hate, and axons especially are particularly sensitive to compression. They hate hypoxia as well, they hate hypoglycemia, but most of all, they hate compression. So that's the main function of the cerebrospinal fluid. The other is waste removal, as the central nervous system doesn't have a lymphatic system. And there's a few others as well. In particular, anyone who's done integrated systems anatomy and physiology will know about its functions in converting hydrogen ions into carbon dioxide to give us a sense of our respiratory efficiency. It is also involved in some buffering systems as well, but I don't want you to worry about that. Buoyancy is the most important one. How much is produced each day? Around about 500 milliliters. So there is a reasonable amount of CSF produced each day. How is it produced? Well, you can see here's a capillary that's going to be sitting in the pia mater. And what happens is that you get the plasma from the capillary being filtered out as interstitial fluid, and then it's filtered again by the choroid epithelium here. So by the time it reaches inside the ventricle that you can see here, it's an ultrafiltrate. So let's have a look at the structure of the ventricles. This structure here is the lateral ventricle. This is the body of the lateral ventricle. This is the anterior horn, and the anterior horn sort of goes into the frontal lobe. The posterior horn that you can see here, this is the posterior horn, goes into the occipital lobe, and the inferior horn here goes into the temporal lobe of the brain. That's important to know when you're looking at cross-sectional imaging of the brain. And then what happens is that the CSF that's produced around here will go into the third ventricle here. So this space here is the third ventricle, and it gets there via this foramen called the interventricular foramen. We'll be looking at that again in the next slide. From the third ventricle, it gets to the fourth ventricle down here via this, and this is called the cerebral aqueduct. And the cerebral aqueduct is actually lying right in the middle of parts of the brainstem. So the third ventricle is between two structures called the thalami, or for individual thalamus, and the fourth ventricle is lying in a part of the brainstem, as is the cerebral aqueduct. That's also lying within the brainstem. Then from the fourth ventricle, it can either go into the subarachnoid space or continue inferiorly in the central canal of the spinal cord. And you'll know what the central canal looks like because we've done the spinal cord now. So how does CSF actually flow? Where is it produced? Well, you can see here's the choroid plexus, and there's choroid plexus in the lateral ventricles, in the third ventricle, and in the fourth ventricle. So it gets produced, it flows from the lateral ventricle out through this is the interventricular foramen. You can see that I've changed it, it's a different font here. This used to be known as the foramen of Monro. So if you see that terminology, don't panic, it's the same thing. So here we are in the third ventricle between the two thalami. Then we go down into the cerebral aqueduct. You can see it's right here, embedded into the brainstem. And then we go into this sort of triangular shaped fourth ventricle. Now at this point, it has a couple of options, 3 in fact. It can either go out a little foramen that's in the midline, and this is called the foramen of Magendie, sometimes called the median aperture, or it can go out in 2 different lateral foramina. Both of those are called the foramen of von Luschka, so we have the right and left foramen of von Luschka, otherwise known as the lateral apertures. If it leaves through the foramen of Magendie, then the CSF is going to go into the cisterna magna. If it leaves through the foramen of von Luschka here, or the lateral apertures, it's going to go straight into the subarachnoid space. So cisterns like this one is just it's a special name for a dilation of the subarachnoid space, and that's what you can see here. Now from here, it may choose to go inferiorly, bathing the subarachnoid space of the spinal cord, okay? Or, as we said before, if it doesn't leave the fourth ventricle, it will continue inferiorly into the central canal. And you may be thinking, well, how does it actually get reabsorbed? And for that, I'll take you right up to the subarachnoid space superiorly. You can see what happens is that there's a little structure called an arachnoid villus, and that's going to protrude into these dural venous sinus, or one of the dural venous sinuses, called the superior sagittal sinus that you can see here. And so the CSF is actually drained into this venous sinus. And so that ensures that we don't get a buildup of too much cerebrospinal fluid within the ventricles and increase the pressure both within and around our cerebrum. So in that way, the cerebrospinal fluid sort of gets recycled, which is kind of cool. So let's have a look at the ventricles of the brain, just as a recap. So let's move it around a little bit. All right, cool, that's a good view. Here are the anterior horns of the lateral ventricles. Here's the body of the lateral ventricle, posterior horn, inferior horn. You can see that it goes from the lateral ventricle to the third ventricle via, let me just see if I can get a better view of this. Yep, cool. Via the interventricular foramen, and there's one for each side. If we come around, there we go, we can see that. Oh, let's just spin around one more time. See if we can capture it. Oh dear. Okay, anyway, we can see that there. There's 2 interventricular foramina there going into the third ventricle. From the third ventricle, CSF goes through the cerebral aqueduct into the fourth ventricle. And then let me just spin this around posteriorly. All right, cool. Then it will either go into the cisterna magna via the median aperture or the foramen of Magendie, or directly into the subarachnoid space via the 2 foramina of von Luschka that you can see there. Or, if it doesn't leave the fourth ventricle, it will go down into the central canal of the spinal cord that you can see there, labelled in green. Ventricles on imaging, what do they look like? Well, here's the lateral ventricle. Here's the third ventricle. This is a mid-sagittal T1 MR, by the way. You can probably tell it's a T1 weighted MR because the CSF is dark. Right, so it goes into the third ventricle. Then you can see it will go through the cerebral aqueduct here into the fourth ventricle. Fourth ventricle, if it goes out the foramen of Magendie or the median aperture, it will go into the cisterna magna. And you can see that will either go down into the subarachnoid space of the spinal canal, or it will go into the subarachnoid space surrounding the cranium. We can't see the lateral apertures on here, and we can't see the rest of the subarachnoid space. Why? Because this is a mid-sagittal slice. We'd have to look at a parasagittal to get that. Right, ventricles and other imaging. Here's a coronal slice, again an MR, T1 weighted MR. You can see the body of the lateral ventricles here, and we can see the third ventricle just here, indicated by 5. Finally, here's axial imaging. And so this is going to be superior, and this is going to be the most inferior cut. You can see part of the choroid plexus here, where my arrowhead is at the moment. You can see the body of the lateral ventricles here. You can see the anterior horn just here. You can see the posterior horn here. So again, anterior horn, body, posterior horn, coming down into the third ventricle there. Cool, that's ventricles.